Hey everybody! So today we're going to learn about observing patterns in inherited traits, so Mendelian and non-Mendelian. So the first thing we're going to talk about is that there is evidence of common ancestry among all organisms. So one piece of evidence is that nucleic acids are in all organisms and they store and transmit genetic information. So that could be either DNA or RNA. And then those both are going to be used to make protein. So all organisms have um, nucleic acids. The other piece of evidence is that all organisms have ribosomes. And if you remember from organelles, ribosomes make proteins. So all organisms have ribosomes. And then the third piece of evidence that we have a common ancestry is that every organism does some type of cellular respiration. So it could be anaerobic, where you don't require oxygen, or aerobic, like our mitochondria do. Okay, so we're going to start with Mendel's law of segregation. So that states that when chromosomes are separated into gametes in meiosis, the alleles are also separated, and that allows for genetic variation. So here you have an allele from one parent, so this would be a homologous pair of chromosomes, so like a big R from mom and a big R or a little r from dad. And in meiosis, those are separated. And so that gives genetic variation because there's lots of possible combinations. So that's the law of segregation. The law of independent assortment is that traits separate independently in meiosis. So, for example, what Mendel thought was that your genes are all going to be independent of one another. However, since Mendel did his experiments, we have learned that there is something called linkage, and that goes against independent assortment, where genes are on the same chromosome and tightly close together. So if you think of people with red hair, they often have freckles, right, that go with the red hair. So that would be two genes that are closely linked. And we'll talk more about that in a little bit. Okay, so just some vocab words. You need to know what homozygous is, right? Homo means same, so that's the same alleles at a locus or location. If you see the word loci, L-O-C-I, that means locations, plural. And so homozygous can be big A, or little a. And then hetero means different. So heterozygous, the alleles are going to be, the genotype's going to be big A, little a. Okay, so a monohybrid cross. A couple more vocab words that you need to either review or remember is phenotype, right? That's the color, or if you have the trait or you don't. The genotype is the alleles. Something you may not have remembered is the P generation is always, it stands for the parental generation, and they're what's called true breeding, which just means that they're homozygous. Okay, dominant is where you only need one allele to make the trait show up. So for example, the big R dominant causes the flower to be purple. And then recessive, you need both recessive alleles, little r, for it to be white. So if you have one big R, right, that's the dominant, it's going to be purple. So the P generation gives rise to the F1 generation. And then if you have an experiment like this, the F1s are mated together, or in this case, self-pollinated. And so your two parents would be big R, little r, crossed with another big R, little r, and that's where you would get this Punnett square. But mono means one, so a monohybrid cross is one trait. Okay, another thing you need to know is a test cross. So test crosses are used to find out the genotype of a dominant individual. So let's say this purple flower here, right? We don't know if it's big P, or big P, little p. So just looking at it, we can't tell. We can tell the genotype of this recessive flower because it has to have two recessive alleles. So what you do with the test cross is you cross the unknown dominant with the recessive. And if the unknown dominant is homozygous dominant, all of the offspring will be purple, okay, and or dominant. And if you cross, and this one is heterozygous, so big P, little p, and you cross it with a recessive, then you'll get 50 dominant phenotype and 50 recessive phenotype. So make sure you write down kind of the results of what would happen when you do a test cross so that you know what that dominant individual's genotype would be. Okay, so a dihybrid cross, and we'll do more of these in class. Di means two, so we're looking at two traits. Okay, so here we have color, so yellow and green. And then our second trait is wrinkled or smooth. Okay, so yellow and green, that's one trait, that's color of the seed. And then wrinkled or smooth is the second trait, which is the shape of the seed. So again, you have your P generation, right? And this is just showing what gametes they're going to give. So now you've got 4 different alleles because each genotype needs 2 alleles because you have 2 chromosomes, one from mom, one from dad. And so this parent, so yellow is dominant, and then smooth or round is dominant, whereas over here, green is recessive and wrinkled is recessive. So those are the gametes they give. All of the F1 generation, their offspring are going to be heterozygous for both traits, which means they're both going to be dominant. So smooth and or round and yellow. And then when you cross one of those, and this is the text over here, if the genotypes of the 2 parents are both heterozygous for each trait, then the phenotype ratio, so what they come out, like the color and the shape, will always be this magic 9331. So in this case, so you would always have 9 dominant trait number 1, dominant trait number 2. You would always have 3 dominant trait number 1, recessive 2, and then vice versa. And then you'll always have one recessive for the first trait and one recessive for the second trait. So that is a really important ratio to remember, but only works if both parents are heterozygous for both traits. And we'll do some dihybrid crosses in class. But go ahead and draw this and make sure that you have this written down. So the dominant for this one would be yellow and round, right? And then 3, so yellow is dominant, round is dominant. 3 would be yellow and wrinkled. And then this 3 would be green and round. And then this one would be green and wrinkled. Okay, so some other things to remember, and this is probably a review. Complete dominance is what we just talked about, where you have one dominant allele is going to cover up everything else. Incomplete dominance is this one. So a red flower and a white flower give you a pink flower. So the alleles blend, the phenotypes of the alleles blend, versus this one, so codominance, I think of co-captains, this is where both of the phenotypes are expressed. So both the red and the white are shown. Okay, so crossing over. We talked about crossing over in meiosis, but now we're going to look at how it affects linked genes. So if you look at these, right, these would be homologous chromosomes. These 2 would be sister chromatids. They're identical, and these 2 would be sister chromatids, which are identical. So you may get a question that says, if you had crossing over, what are the possible allele combinations? So you could get, and remember in meiosis, you only get 1 chromosome because you get half of the information. And so a possible, if these 2 crossed over, you could get big A. If it crossed over right here, little b, little c, you could get little a, right, big B, big C, and you could, if it crossed over kind of twice, you could get big A, little b, big C. Okay, and another kind of way that they talk about this is that genes that are tightly linked are ones that are closer together, right? And there's a less space for crossing over to happen there. Like it would have to be right in between here. Whereas between A and C, there's lots of room. And so A and C aren't going to be as tightly linked or travel together. So something like red hair and freckles would be more like the B and C gene, right? So crossing over isn't as, doesn't happen as often. So those are the ones that are not closely linked are going to be like A and C or A and B. So go ahead and draw that and kind of make a note of what tightly linked and not tightly linked are. Okay, so we're going to do a problem. So I'm going to explain it the first time. So you're definitely going to want to write this down in your notes. And then there's going to be a couple more problems where you're going to pause the video and try the problem. So you could be given the frequency of crossover, okay? And you're given the frequency of crossover between these different genes, and what you're going to need to be able to do is put them on a map, on a chromosome, and show relative position of them. So the first step is you're going to start with the genes that are furthest apart. So if there's more crossing over, then that means that they're further apart. So if you look here, 45% is the biggest, right? And so B and C are, if they have 45% frequency of crossing over, that means that they're the furthest apart. Okay, so you put those, and like the directions are right here. So start with the genes that are furthest apart. B and C are 45 map units apart. So look at this, map units is the same as saying percent crossover. So you put B and C, and see this little bracket thing, 45. And then you go to the next biggest one. So B and D is the next biggest one. So you know that B and C have to be 45, and so D has to be on this side, which is 40. And then you just kind of solve it like a puzzle. And then just make sure everything, so then you go to your 30, A and C, right? And then A and D is 25. Now, to figure out how many map units they are, you're going to have to do some subtraction, right, to find the distance between the genes. So what that means is, so to find this 15 MU or map units, they took 40, which is this whole distance, minus 25, which is this distance, to get 15. Okay, you already know this is 25 because it was told in the problem. And then for this one, you know that this is 30 between A and C because of this right here. And then between A and D is 25, so 25 plus 5 is 30. So it's just kind of simple math, little puzzle, kind of fun. And so the ones that have the most crossover between them are going to be the furthest apart, and the ones that have the least amount of crossover between them are going to be the closest together. Okay, so we're going to do this one as a task, and then I want you, and we'll explain it, and then you're going to try a couple on your own and pause the video. All right, so you may be given a problem like this. So it says, assume the cross is made between one heterozygous plant and one recessive plant, which is kind of like a test cross, except you wouldn't know this one. And the offspring are going to give you these numbers. So you get 4 different genotypes of offspring. This number is the number of offspring, okay? And you want to find how many map units are there in between the A gene and the B gene. Okay, so when you see a problem like this, you want to think about what are the parental genotypes. So what are the genotypes that are just like the parents, and then what are the recombinant genotypes? So recombinant genotypes means that there was crossing over. So they're different than the parents. So you have a heterozygous for both traits here, and then you have homozygous for both traits. So this one is a parental genotype, and this one is a parental genotype. And notice that the parental genotypes are always going to be bigger than the recombinant ones. Okay, so in order to figure out how many map units are between the A and B genes, you take the 2 recombinant numbers of how many offspring, so 48 and 52, and you divide it by the total number of offspring. So if you add up 106, 48, 52, and 94, then you get 33, and so it's, or 0.33, and you turn it into a fraction, and so it's 33% recombination between them, and so they're 33 map units apart. Okay, so make sure you write down that example so you have a good example. Okay, so this one, I want you to pause the video and see if you can do it. Okay, so the answer to this one is you add the recombinants. So it would be 130 plus 120. Those are the two recombinant phenotypes. So you can do it for phenotypes or genotypes divided by the total, which is 1,000, and you get 25% recombination, which translates into 25 map units between those two genes. All right, here is another one. So go ahead and pause the video. So on this one, you're looking for bristle type and eye color, okay? Oh, the other thing I wanted to tell you was that whenever you see wild type just means kind of the natural, what it is in nature, and these are dominant. Okay, so wild type bristles are going to be dominant, and wild type eyes are also dominant. And so the opposite, the mutation, would be the scute bristles and the ruby eyes. Okay, so in order to find this one, you take the recombinant numbers, which is 188 and 192, divide it by 1,000, and you get 38% of 38 map units. These were the parental phenotypes, and these are the recombinant phenotypes. And that's it. We'll do some of these problems in class, but make sure that you have a good example or two written down. And that's it. Hope you have a great day.